Okay, well, good morning and good afternoon, everyone. I'm Brian Felton in the exobiology branch at NASA Ames, and it's great to see you all here for our fourth presentation in our AI seminar series. Before we begin, though, I just wanted to take a moment to highlight the uniqueness of our next two presentations. If you've been following us from the beginning, then you've seen talks given by researchers in astrobiology that have applied AI/ML to their own work. But this two-part education series is a departure from those seminars and serves as a way to address the need to improve the community's general knowledge of AI/ML. If you stop and think about it, AI/ML is a fairly complex and layered field when it comes to the vocabulary and techniques, especially when you consider neural networks and deep learning. And many of us, including myself, don't have formal backgrounds in data science. And so the intent here is that by becoming more familiar with AI/ML, the astrobiology community is better positioned to take the next steps towards research paths and collaborations involving AI/ML. And so with that said, I'm very pleased to introduce Dr. Floyd Nichols today from the Department of Geosciences at Virginia Tech. Floyd specializes in geochemistry, biosignatures, and data science, and will be the presenter for this two-part education series. Today, he's going to be focusing on unsupervised learning techniques, and the second part will be held in 2 weeks on September 24th. So Floyd, again, thank you very much for being here. Please share screen, and the floor is yours.
Thanks, Ryan, for the introduction. I'm really excited to talk a bit about unsupervised learning today and in 2 weeks supervised learning. So hopefully you all learned something new here. Just to make sure though, can you see my screen?
Yes, it looks good.
Okay, great. Yeah, so today is a gentle introduction to machine learning, part one, unsupervised learning. And before I get into the presentation, I'd like to start off with sort of a fun interactive activity, more specifically a word cloud. And so in the top left, you'll see a QR code. And if you use your phone, or you can also use your computer if you're on your computer, I just want you to take a minute or two and think about some of the AI ML techniques that you know about. And this could be anything, such as, ChatGPT, principal component analysis, etc. Yes, a lot of PCA, deep learning. Very interesting. So it seems like a lot of people are more familiar with perhaps some of the supervised methods, regressions, linear regression, random forest, k-means. Awesome. Well, thanks for anyone who participated here. So we see here, we've got a couple of different options from PCA to random forest to regression, which can mean a lot of different things, linear regression, k-means, deep learning. And so what's kind of exciting about this is there are many, many more techniques available for AI ML. And so hopefully over the next 2 weeks, or I guess 3 weeks, you guys will learn some of the different methodologies that you can employ for your own work. And so now that we got that out of the way, what exactly is machine learning? And why might we be interested in using it for astrobiology? Well, there's many reasons. One being that machine learning is about designing algorithms that automatically extract valuable information from data. And when you're thinking about your own work, especially in astrobiology, we're often attempting to extract information about complex systems. And there are a variety of reasons for why we might want to use this for astrobiology outside of extracting valuable information. And one of those is actually to help drive machine learning, research forward as well. So inherently, machine learning is data driven. But with that, there is often a lack of domain knowledge for machine learning. But with recent work, let's say within the last 5 years or so, there's been a paradigm shift towards knowledge-informed machine learning or theory-guided data science. And so us astrobiology researchers can play a critical role in not only improving our own work, but also improving machine learning methods as well. And so finally, as I mentioned before, within the last decade or maybe even 2 decades, astrobiology data has become extremely complex and or very large. And so we've also, like many other fields in STEM, have entered the realm of big data. And we need really, we need important methodologies and efficient methodologies to actually extract information. And that's where machine learning comes in. Um, and so you often hear AI and ML together, artificial intelligence and machine learning together. But really, what does that mean? What is it from a broad sense? Well, we can think of artificial intelligence as an interdisciplinary field that involves mathematical statistics, information theory, game theory, optimization. Sometimes it's only one of those aspects, sometimes it's a combination of multiple, and sometimes even all of them. And we can think about artificial intelligence in, let's say, a web or branches. So artificial intelligence is actually just an umbrella term to denote multiple other branches. One, which is machine learning, which we often talk about and what this presentation is discussing today, but you also have robotics and planning, natural language processing, which many people might be familiar with, expert systems, speech, and even computer vision. And so there's many aspects of artificial intelligence, even though we will only focus on machine learning today. And even from here, you have even more branches within these subfields. And specifically machine learning, there is supervised learning, unsupervised learning, semi-supervised learning, reinforcement learning, as well as deep learning. And some of these concepts people may be familiar with, and as the word cloud has suggested, a lot of people are already familiar with deep learning and some aspects of supervised learning as well as unsupervised learning. But one thing I want you to take away from this figure is, the, what I'll describe today and in 2 weeks is only a small fraction of what artificial intelligence actually is. And so that's really exciting, I think, for the field of astrobiology as well as machine learning, because there's many, many avenues that you can take to sort of improve your own work in also the field of data science and machine learning. Today, though, as I mentioned, we will focus on unsupervised learning. And so let's take a look at what that actually is. Unsupervised learning essentially is a method or a branch of machine learning that uses clustering of unlabeled data sets to essentially discover hidden patterns or data groupings without the need for human intervention, hence unsupervised. And again, I'll discuss this in 2 weeks, but supervised learning will use some sort of human intervention. And so one of the powerful aspects of unsupervised learning is the fact that you don't need that human intervention. And so there are a variety of use cases that we can employ for unsupervised learning. And this isn't all of them, but these are definitely the main branches and probably the most important ones for astrobiology. And that being dimensionality reduction of your data, clustering of your data, as well as anomaly detection. And so this is where unsupervised learning becomes extremely powerful. So we'll first start with the first use case, which is dimensionality reduction. So what exactly is dimensionality reduction? Dimensionality reduction is a method for representing a given data set using a lower number of features while still capturing the original data's meaningful properties. And so this can sort of, if you're not familiar with data dimensionality reduction, it can be sort of esoteric when you're thinking about it, especially since humans can only say, visualize or understand three dimensions. But with many data sets nowadays, in the modern, we're often working with multiple variables or multivariate data that extends well beyond three dimensions, sometimes upwards of hundreds of dimensions, and in some cases even thousands of dimensions. And so, how can we best represent that data in a more palatable way? And so here's a sort of rudimentary example of how we can think about dimensionality reduction. And again, because humans can really only visualize three dimension, this is sort of the best representation that we can give. But on the left side, you see two cubes, um, a larger one, the blue one, and a red one, the smaller one. So when we're applying dimensionality reduction methods, we want to capture the essence of that higher dimensional data. So as we look on the left side with three dimensions, we can reduce it to, say, just one of the surfaces of the cubes. And what we see is, okay, we still have this larger surface from the blue cube and a smaller surface for the red cube. And then from here, we can reduce this to one dimensions. Well, actually, this is two dimensions, but we can reduce this further, and we still see, okay, we now have a blue line that's large and a red line. And so from this data set, we have a lower dimensional representation of our high dimensional cubes. And I'll show you later in this talk how we might actually represent that in actual data. But this is essentially the most basic form of what dimensionality reduction is. And so there are many methods, and as you guys had already put in the word cloud, standard PCA is probably one of the most popular and most well-known methods for data dimensionality reduction. And as I mentioned before, these days we're working with multivariate data, and we can think of this as a cube where a data point is within a multi-dimensional space, in this case, three dimensions. And so if we want to preserve the structure of this data while also finding patterns within the data, we can reduce this three dimensions to two dimensions. And we can see here that, okay, these different colored points are still grouping together even though it's in lower dimension. And we can reduce this even further to, in this case, one dimension, where we still see most of the patterns within this data set. However, we can see that the two dimensions probably the best representation of this higher dimensional data set. And so the way that a standard PCA works is it's essentially linear algebra. It uses a set of linear transformations to preserve the global structure of your data. Because this is a linear method, and you'll notice this with the other unsupervised methods that I discuss, as well as when we get into the supervised learning portion of this series, is that linear methods are often very, very fast to compute, as well as easy to interpret. And so that's one of the major pros of utilizing standard PCA. And here is just a sort of visual representation of what is actually going on in a standard PCA. And again, this is not something that one can visualize extremely well because of the fact that this is transformations from high dimensional data. But what you can see here is a line that's rotating along an axis. This is essentially represent that linear transformation of your data. And so essentially what the PCA is doing is it's looking for some component or principal component that best represents the variance of your data. And so we can see here as this line is rotating, the different points are some distance away from that line. And so we want to minimize this variance with respect to a principal component. And depending on how many dimensions are in your data, you can perform this transformation for as many variables as there are. So in this case, because this is two dimensions, we see one line, hence one transformation. But say we have 20 variables or 20 dimensions, this process would occur 20 times until you get something that is represented in either, say, 2D space or one-dimensional space. So most people are familiar with the standard PCA, but unfortunately, for most astrobiological data, or really geoscientific data, is often nonlinear. And so to use a standard PCA, you actually have to make some assumptions about your data, that being that your data is linear in nature. But because we know this is not the case for most astrobiological data sets, a standard PCA is not always the best method to use. And so there are alternatives if we want to maintain the global structure of our data as we reduce its dimensions, that being a kernel PCA. And unfortunately, there is not a great representation visually that describes the process of kernel PCA because of the fact that it uses nonlinear transformations. But if you are interested, I can answer questions later. However, um, for many of the kernel PCAs, you're using some sort of polynomial transformation or what's called a radial basis function. And essentially what that does is it's a bit counterintuitive, but you take your high dimensional data, you throw it in, you map it into a higher dimensional space where it can be separated by, say, some hyperplane, and then you remap it to a smaller dimension, or say 2 dimensions or even 3 dimensions. And so it's a bit counterintuitive, so I won't describe the process too much, but I'd be happy to answer any questions that people might have with kernel PCA. But the main thing to take away is if you're working with nonlinear data and you want to reduce the dimensions, then kernel PCA might be an option. However, maybe you want to reduce the dimensions of your data that is nonlinear, but you are interested in, say, things that are not global structure. Perhaps you're interested in local relationships. And a method that has gotten a lot of attention recently is the t-distributed stochastic neighbor embedding, or t-SNE for short. This is slightly different than a PCA in the sense that a PCA is using the linear transformations as described in the previous slide. However, with t-SNE, or t-distributed stochastic neighbor embedding, what it's doing is it looks at a point of interest, it looks at its neighbors, and it then calculates some probability distribution that a point is similar or dissimilar to said point of interest. And so, unlike the PCA, t-SNE is great with nonlinear dimensionality reduction. However, it's computationally more expensive because you have to look at each point individually, look at its neighbors, look at its similarity and dissimilarity, and calculate a probability distribution for each point of interest. So it is very valuable when you're attempting to look at this nonlinear data. However, it is important to keep in mind this computational expensiveness, as well as it's a slightly less easy to interpret. The other thing with t-SNE, as opposed to PCA, is PCA is very straightforward. As I mentioned previously, it's a lot easier to interpret than these nonlinear methods because it's just using that linear transformation. However, and you'll see this a little bit later in the sort of Python walkthrough, but t-SNE actually has multiple hyperparameters. And what that means essentially is you can get a different clustering of your data based on, say, some parameter for your actual dimensionality reduction method, in this case, t-SNE. And so one of the major important components of t-SNE is this value called perplexity. And so you can see in this GIF, as we increase or decrease the perplexity, we get differences in the form of clustering. And the primary reason that is because perplexity is describing how many neighbors are present for your point of interest. So the more neighbors you have, the more clustering you'll have. or the tighter clustering you have, and then the less neighbors you have, then the more spread out you'll see the data. And so very powerful, but it is important to keep that in mind. And so there are many other different types of dimensionality reductions. Here I'm just describing PCA, one standard PCA, kernel PCA, as well as t-SNE, but there are a variety of other methods that one can use, and perhaps I can share with Ryan a list that he can send out. But there's maybe over, 50 to 100 different dimensionality reduction methods, but these are definitely the most common ones and probably the most applicable to astrobiological data. And so in the second use case that I described for unsupervised learning, there is clustering methods. And so clustering is slightly different than data dimensionality reduction in that clustering is classifying different objects, data points, or observations into groups or clusters based on similarities or patterns. And so similar to the dimensionality reduction, there are many, many different types of clustering methods. I'll obviously not go through all of these, but here you can see different colors representing, say, different classes. And these are all determined based on some algorithm. And going forward here, I'll just discuss two very popular ones that one which is really good for linear data, and then one which is good for nonlinear data. And so the first being k-means clustering. Um, this is one of the most common and probably one of the most powerful clustering methods actually for unsupervised learning. And what k-means clustering does is you can see these different x's for data points, and these x's represent a centroid, and that centroid represents one particular class, hence why there's many different colors. And as we increase the number of iterations of this algorithm, you can see here epoch number, which is just an iteration. As we increase the number of iterations, we improve the average values for each centroid until there's no convergence. And so when we get to, say, epoch 10, you'll notice that all of these centroids are roughly in the center of these different clusters. So k-means is actually very similar to principal component analysis. And what I mean by that is very simple, it's computationally inexpensive, so it's very fast, and it has much easier interpretation because it's using linear methodologies. However, one of the biggest drawbacks for k-means clustering is that the clusters must be predefined and can only identify spherical clusters. So you can see here, as you look at the centroids, you mostly see these spherical shapes around the colors. And so, again, when thinking about astrobiology data, we might not always be working with data that is nice and spherical, like what we see here. And additionally, we might not know how many clusters we actually want. And so another method that can be employed for understanding patterns and clusters of data is dbScan. dbScan is a little bit more complicated than k-means. So k-means, you look at a centroid and you see how many points are close to that centroid, and that becomes its classification. dbScan, on the other hand, has a random iteration of a point, and based on some parameters, so actually very similar to our t-distributed stochastic neighborhood embedding where there are hyperparameters, we will look at a core point, see how many points are close to it, and depending on how we define how many neighbors there should be, we'll classify that as a cluster. And so that's a little bit, let's say, uh, difficult to understand perhaps. And so here's a much better representation of what is happening with dbScan. So you can see here, we look at a point randomly, and we sort of walk along the each point based on the hyperparameters chosen. In this case, epsilon is 1.0, which is the width of the circle, and then the minimum number of points is the minimum number of neighbors, essentially. And so you can see here, all of these points are 4 points, which means that they all have a minimum of 4 points for neighbors within that epsilon one. And anything outside that is then defined as a new cluster. Um, what's really powerful about dbScan compared to k-means is that the number of clusters does not need to be predefined, which is which is a plus when you don't know uh, the structure of your data. Uh, and you can see here, unlike the k-means algorithm, there are some spherical shapes, there are not some spherical shapes. And so when you're working with data that has these weird ambiguous shapes, dbScan can be very powerful. Additionally, um, it can tell you somewhat about outliers, which are some of these points out here, which are not colored in the final instance here. However, like t-SNE, anything that's nonlinear is almost always going to be computationally more expensive. So dbScan, very expensive, can take a lot of time if you're working with large amounts of data. And it also requires hyperparameter selection. So it's very sensitive to that. However, if you can determine your hyperparameters well, then you can actually uh, get something really, really powerful. And another method that I won't describe in detail for this introduction is autoencoders. And the primary reason why I won't describe this in detail is because this is a little bit more advanced concept that requires some knowledge of neural networks. However, I know with a lot of astrobiology data, people are starting to use neural networks more. And so I figured that I would introduce that concept here for those interested in. And so autoencoders are sort of a special type of unsupervised learning used for neural networks. The main application is essentially to capture key components from data within neural networks. And so the best way to think about this is say we have a picture. In this case, perhaps we've got some cute cat and some cute dog or something like that. And the neural networks find some hidden patterns within this data set, these images. It then sends them to lower dimensional space, which we call latent space here for neural networks. And this is actually very similar to PCA and t-SNE, but from a neural network perspective. And then it reconstructs an image based on this latent space. And so the reconstruction might look like a cat, it might not look like a cat, it might look something very similar to the original image, but not exactly the same. So it's really great when you want to capture these complex features from images, say you're working with remote sensing data, for instance. But really, with all neural network, anything related to neural networks, very computationally expensive and can result in overfitting sometimes. And so um, you really have to hone your hyperparameter selections down for these as well. So there's many toolboxes available for implementing machine learning in your data, mostly using the Python programming language or R programming language. There's libraries such as Tidy Models for R, scikit-learn for Python, PyTorch for Python, Torch for R, TensorFlow and Keras for both R and Python. And so these toolboxes can be used for a variety of different things. Specifically, tidy models and scikit-learn are very similar, just that tidy models is for R, Python is, or scikit-learn is for Python. And these are great if you want to employ, say, unsupervised methods like the ones described today and later in 2 weeks when I described some of the supervised learning, classical supervised learning methods. These are really great for that. And Torch, PyTorch, TensorFlow, and Keras, these libraries are going to be much better suited for developing and building neural network models, but we won't describe any of that today. And one of the important things to note with these toolboxes is, well, as most astrobiologists, you probably don't want to employ math. And so the nice thing with these toolboxes is they already have these predefined functions that don't require you to have any of the mathematical knowledge, although it is important to have some background in the math knowledge, but you don't need to actually know how to program the math to actually do it. So it's these nice, neatly packaged functions that allow you to get things done. And so I'll show you a little bit of how one might actually implement this in their own data set using a Python code walkthrough, and I'll be using scikit-learn for this actually. But if you don't use Python and you much prefer R, then you can use tidy models as well. And so one thing to preface is that here I'm just going to show you mostly code blocks for how to actually implement this. I won't describe all of the code in detail, and I'll actually skip over a couple of the coding aspects just to make this a little bit more palatable. But if anyone is interested, I believe there should be a link available from this seminar where I've developed a publicly available Google Colab notebook if you want to run and play with this code a little bit more. And so the first thing is you'll actually load in your libraries, but there's actually a lot of different libraries that you use here for this particular uh, setup. And so I'm not showing you that just because I don't want anyone's brains to hurt. And so again, if you do want to look at every single library and module of used for this, and you can look at the coding notebook. But one of the first things that we'll do when you want to examine your data specifically for unsupervised learning is we'll first import our code. And in this case, I'm actually showing data from a recent publication, which is also available on GitHub. But we'll pull in the URL, which is just a CSV file of this data. But if you have your own data, you can also import your own CSV. We'll use a function here called read CSV, which allows us to extract all the information in table format or data frame format from the CSV file. And then here's just some cleaning up of the data. So getting rid of any NA values that we don't want. We'll look at a preview of our data using this head function, and we'll denote an X, which is essentially just a matrix for our unsupervised learning. So after after we clean up our data and we use this head function, we can see sort of the contents of this data. And here, for this particular work, where it's actually total organic carbon with respect to different elemental abundances. So you can see here sample total organic carbon percent and the counts of the elemental abundances that I use for this data set. So after the data is loaded, we're actually going to do a preliminary test, something I didn't describe previously, but here we're actually going to assess the multicollinearity of our data. And one reason why we might want to do this is just to get a sense of um, how well or how much we should trust, say, linear methods. Linear methods are going to rely heavily on data that is not high in multicollinearity, and that is primarily because the higher the multicollinearity you have, the more your parameters actually break down, and so it's a little bit hard to assess. But this actually becomes a little bit more important when thinking about supervised learning. But this is just preliminary work that we'll use. Oops. And so to calculate multicollinearity, we can actually use a function called variation inflation factor. And that just is a way to relate your R squared. So a high R squared of 1 equals essentially extremely correlated data. And then anything less than 1 indicates less collinearity. There's actually a more complicated equation that goes into this, but that's the best way to describe it. And so here we'll convert our variation inflation factor into a data frame, which is what this function is here. We'll then describe our features, which are just our columns. So if we go back here, our columns, magnesium, titanium, vanadium, we'll just describe those as features. And then we'll run sort of an iterative function here or for loop that calculates our variation inflation factor, which is this function here for each individual column. And what we get is we can plot this and take a look at it. Um, the dashed line represents a number that is generally considered to be high multicollinearity, which is about a value of 10. And we can see here that for this particular data set, most of our features are extremely multicollinear, have extreme multicollinearity. And so, as I mentioned before, this can pose challenges for our model interpretations, specifically linear models. And so this is just important to keep in mind. But this is just kind of our first order process that I think is really important to use and implement before assessing the rest of your data. And so now we can then use our scikit-learn to reduce the amount of dimensions in our data and also do some clustering as well. And so we'll do that here. And it's actually very simple. You'll see here, this is just some cleaning up code. Essentially, we're going to set our matrix, our X matrix, which is all of the values from our columns for the elemental abundances. We'll do some normalizing here as well, just to make things run faster. And if you are working with, say, data that has different units, then normalizing is actually going to be really important. And then here we'll also describe just a Y variable or a response variable for coloring. Um, and what's really nice is when you use scikit-learn, it's really just one function that you have to use to use a standard PCA. So here we're going to denote the number of components, which is really just the number of dimensions that we want, in this case two. So we're going to take our extremely large data frame that had over 30 features and reduce it to something that makes a lot more sense, which is two features. We'll use this PCA function. We'll put the number of components in. This is just telling uh, the function how to actually reduce our data. This pipeline is just a way to implement our normalization and scaling factor. And then we'll transform it using those linear transformations. So this fit.transform function is going to then implement the linear algebra. And so once we do that, we'll also describe our k-means clustering and dbScan. Again, just one function, which is very nice, our k-means function. And as I mentioned before, k-means, you have to denote the number of clusters. And so we'll just, in this case, we'll just say 3 clusters just because that's just usually a good starting point, but you'll know your data a little bit better. Random state indicates if you're familiar with seeds and modeling, you're always starting with a random initialization. And if you want it to be reproducible, you should set some random state so that the initialization is always the same. And then similarly with dbScan, we'll use this dbScan function. We'll indicate the hyperparameters epsilon, which is the diameter of the circle, minimum number of samples, which is your neighbor, and then we'll fit that as well. And then we can actually do the same process with tsne, but rather than use PCA function, we'll use tsne function, and we'll have to implement some of those other hyperparameters that I mentioned. But everything else stays the same. And so one of the nice thing is when we do this, we can actually see this now from a actual real data perspective. And we can see that when we reduce the dimensions of this total organic carbon versus elemental abundance data, we see very different clustering. And so with PCA, we see mostly 3 main clusters, one being the top, bottom left, bottom right, whereas tsne, we see much better clustering and more clustering as well. And so we can then label that, say, by total organic carbon, and we can see a little bit more clearly what this actually looks like. We can see that total organic carbon, that's about 6 to 9% for PCA is roughly in this area. We've got a little bit more variation here in this bottom left cluster, but we've got about moderate levels of total organic carbon as well as low levels. And then in the bottom right here, we have mostly low levels. And one of the nice things is when we look at the tsne labeling, we get much better clustering of that, especially this variation here that we see in PCA. So this might indicate when we employ both of these methods that the nonlinear clustering was better, which might indicate that our data is also very nonlinear, which makes sense when you're thinking about hypersaline lakes, which is where this data came from. So then maybe we don't want to cluster it by total organic carbon because maybe we don't have those values. Maybe we'll want to actually just look at it from a more unsupervised perspective, which implements k-means clustering. So this is using the clusters number 3, as mentioned before. And what you can see here is, okay, it actually sees about the same things that we see from identification of our total organic carbon. And we see that a little bit more, a little less clearly here in t-SNE compared to when we label with total organic carbon. But you can see here, as I mentioned before, with k-means, you're going to have more spherical shapes. Um, but when we do DB scan, which is very nice, we don't have to define the number of clusters. We just have the number, the other hyperparameters. We can actually see something that's more what we might see with the human eye, as well as what we see here as well from just these circles that I did manually. And what you can see here is we actually now see that there are different clusters from our PCA. There's actually 4. And then t-SNE also sees 4, very similar to what we denote here. And dbScan also is really great at uh, detecting outliers. So these black points are actually outlier points from these clusters. And so we get a little bit more information with dbScan and t-SNE together. And so this really shows how powerful using nonlinear methods can be when you're working with these extremely nonlinear data. But, that's just one example and a sort of more of a simple example. But if you're interested in learning more about how some of these unsupervised learning techniques are employed in astrobiology research, I recommend some of these papers here. Unfortunately, unsupervised learning is, a little bit less popular than supervised learning at the moment. But so there's not as many papers, but I think they're still very important and can be really great if you want to take, say, an agnostic approach, such as this first paper, but a lot of these papers are actually sort of describing these agnostic insights, which unsupervised learning can be really useful for. And so with that, I'd like to, thank Ryan and Caleb for giving me the time to uh, go through some of the foundations and fundamental aspects of the unsupervised learning. But also, I wanted to mention that I'm currently working on developing a community tool for hopefully making these concepts a bit easier to understand in an interactive format. It's not currently finished, but if there is interest from people in the community, then I'll definitely try to perhaps pre-release it or something to Ryan and Caleb, and you guys can take a look at the unfinished product if that's something that people are interested in. Yep. So yeah, thanks for listening, and if you have any questions, feel free to put them in the chat or oops, we'll save that for now.
Awesome. Thank you very much, Caleb. Or sorry, for Floyd. Floyd, did you want to do any Q&A before the Slido or?
I'll do Q&A before just to give people time for questions, and then we can save the Slido
Okay.
for last.
Okay, perfect. So if anyone has questions, feel free to raise your hand or post it in the chat, and we'll just go from there. And okay, perfect. Jennifer, go for it.
Sorry, I couldn't, as usual, couldn't find the unmute button. Just a quick question. I know it's on Collab, but I just wanted to make sure I can share this with other people, my students, so forth and so on, and just point them to your site.
Yeah, definitely. So it's available for anybody to use. So feel free to share it with anyone that you think can find use out of it.
Thank you.
Mhm.
And actually, Floyd, I had a quick question. Let me get this, and then I'll get you, Caleb. If you go back a bit, there was when you were talking about the spherical aspects of the clustering data, I think it's even before the collab. You were mentioning, and
This year?
yes, this one, yeah, the k-means clustering. I was a little confused. Are you just saying like what what again was spherical in this part? Because I guess when I'm looking at the data, it
Mm,
looks like it's kind of spread out in little
Yeah,
bunches, and so I was just a little confused.
so I guess that's a great question. What I mean is sort of the classification of the different points. So for instance, this is probably the best example to look at here. This color, which I'm not entirely sure what color that is, but this centroid indicates that any points within some distance to this centroid will have that classification. So you'll see here that these data points from this cluster above are part of this centroid as well as parts in this cluster below. Um, and that's because the radius of this centroid looks like this. However, if we were to actually cluster these data based on, say, density and not just spheres, this cluster would actually be separate from this cluster. And visually, with the human eye, we can actually see that. But with k-means, it can't actually detect that this cluster is different than this cluster. It thinks that these points are the same as these ones, if that makes sense.
Yeah, no, that does. That's better. Thank you, Floyd. Caleb, go
Mhm.
ahead.
Yeah, thank you, Floyd. This is really, really wonderful deep dive, and I think incredibly valuable, certainly to someone like myself who is dabbling in these tools. I have a really sort of basic, stupid question, but I'm interested in sort of scalability. So I was wondering, for example, in your elemental abundance data analysis, when you
Mhm.
run DB scan on that
Mhm.
data, what are you running it on? How long does it take? A really stupid question, but I'm curious.
Yeah, no, that's not a stupid question at all. That's a great question. And so when I'm doing both of these clusters on for both dbScan and for k-means, I probably should have mentioned it a little bit more clearly, but I'm actually clustering it based on the data dimensionality reduction. So in this case, the PCA, and on the bottom case, the t-SNE. The reason why I say that is because you can use k-means or dbScan for data that is maybe you're just looking at the relation between x versus y. It doesn't necessarily have to be dimensionality reduction data. So that's what I'm doing the clusters on. Um, and then in terms of computational time, fortunately for my data set, which is only for this total organic carbon at least, only about 200 data points, and there was 34 different elemental abundances. So in the world of machine learning, that is quite small. And so these clustering methods, both k-means and dbScan, were less than probably zero point 0.5 seconds to run. But say you're working with 100 variables, or with a lot of astrobiology data sets nowadays, you might have over 1,000 samples. Well, dbScan will maybe take a lot longer than k-means. For instance, k-means, because linear, will probably still, even with higher data, it scales very nicely. So the time it takes to compute is still very short. Whereas dbScan, the more data you have, I don't think it's exponential growth, but it's pretty close to exponential. So it gets much slower the more data and the more dimensions you add.
Great, thanks. That's very helpful.
And unfortunately, that's the case with most nonlinear methods, is even though they can often give you a lot more insight into your data, uh, their scalability is a little bit worse than the linear method. So things do take a lot longer to run. And that's one reason why I do like, I'm not paid to advertise Google Colab, but it's quite nice because say you have a student who wants to implement these methods, but they don't have the greatest laptop or computer. Google Colab gives 16 gigabytes of RAM for free to run. And so for a lot of data sets right now, that's pretty decent. Maybe if you want to run neural networks or higher data sets, you might need like 32, 64, or high performance computing. But I think for a lot of what graduate students are working on right now, perhaps even undergrads. Uh, 16 gigabytes is enough that, it won't take a whole data run, but maybe just like a couple minutes or something.
Jennifer, go ahead, please.
So kind of actually, coming off from that and branching off from that, I would love to have your feedback, deploying in the cloud, right? So,
Mhm.
we're entering this period where, data sets, especially, already, right? Thinking about like DES and DECAM and so forth and so on, but with LSST, so forth and so on, even though they have beautiful tools, they have a Rubin Science platform, it still would be nice to have your viewpoint on, um, you know, um, which tools potentially, I kind of already heard it in your response, but which tools potentially play best in the cloud, which ones would require, different HPC, considerations like you're saying. And then of course, deploying on AWS, right? It can be complicated, right? And you have
Mm.
this tier-based system. Yeah, if you have any feedback on that, that would be lovely. And then, or if you wanted to include that in your notebook, that would be great. Because I think about all the time, not just about tools for my students, right? But then for
Mm.
colleagues, and now that we're gonna, have these huge data sets, right, coming through, yeah, there's no way to work other than in the cloud, right? And so
Yeah.
Collab gives you nice stuff, but of course you have to have a Google account. So then,
Mhm.
and so that, yeah, there's other tools like Binder and, Voila, right? Yeah, if you have
Yeah.
any thoughts on to which plays well with what, and yeah, I'd be very curious. Thank you.
Yeah, I think that's a great question. In terms of what plays well with what, I think, we're in this age where, if you look at a lot of recent papers that are not necessarily astrobiology, but just in geoscience in general, a lot of machine learning is being implemented in work just because people are starting to find the value in it. And you'll notice that the most common method right now is deep learning. Um, However, not every problem needs to use deep learning or these really complicated methods. I mean, even here, if say we didn't use t-SNE, we just use PCA, we can still see, almost as much information. We see slightly less, but almost as much. And so, in terms of compatibility, I would say that linear methods are always going to be the best. And so, if you can describe your data using one of the linear methods, those are going to be the most compatible. So hopefully, uh, as people implement and so actually that was one reason, and you'll see this a little bit later in the supervised session if you come, but assessing things like multiclinearity, correlations, there's other different aspects that you can test as well, but this will give you some insight into if you should use linear or nonlinear methods, but most people these days, not that there's anything wrong, but they just tend to go for these more complicated methods. And then in terms of deployment, um, yeah, that's a great question. Uh, AWS is good. It is very expensive though, but I would say AWS is definitely the best if you do say have to use deep learning or these high computational methods, because even though it is quite expensive, they actually do offer a lot of uh, computational power, significantly more than anything that Google gives you. And then Google Colab, so what I mentioned before, the 16 gigabytes is free, but there are also Pro Google Colab that allows you, I can't remember the actual number, but I think maybe it gives you 32 gigabytes or something like that. So maybe you're working with a little bit more data than what the standard Google Colab would be useful for, but you're not working with a large data set that's very complex, which might be better for AWS, for instance, then I would say Pro Google Colab is good. And then in terms of things that are not Google or Amazon, um, I'll have to, I can't remember them all off the top of my head, but I can get back to you and send you a list in an email. But unfortunately, most of them are going to require some account of some sort. Does that answer your questions?
No, that's perfect. And thanks for just coming up with that off the fly. And yeah, I'll be looking forward to your next session. Thank you.
Great. Thanks for the question.
Hey, Floyd, I know we're essentially out of time. Could you try to hit Angelica's question real quick in the chat? Are you able to see the chat?
Yes. I
Okay,
cannot see the chat.
You cannot. Okay, so she said PCA and t-SNE axes are equivalent. How do you compare them?
Oh, that's a great question too. So they're actually not they're not really equivalent. Well, they're similar, but the PCA axis is actually going to be uh, a principal component. Whereas t-SNE, these axes are a little bit more arbitrary, but essentially they are just some represent some dimension. So t-SNE1 just represents the first dimension of your t-SNE, and then t-SNE2 is the second dimension of your t-SNE. And so they're not exactly the same, but they are similar. And then in terms of comparing, the primary use for these dimensionality reductions is to look at clustering, for instance. And so in this case here, I wouldn't necessarily compare, -5 to -40. That doesn't really mean anything. They're kind of arbitrary numbers in some sense. But I would compare the clustering. And so in this case, it's a little bit easier to identify in this data set that I gave here. Sometimes it's not as simple. And so you'll actually, if you do access to Google Colab, you can see this a little bit more. But one of the better ways to quantify this is computing, say, a silhouette score, and that tells you how good of clustering each method is. And so you can compare the silhouette scores of each dimensionality reduction to each other. And so in this case, I can't remember the number off the top of my head, but I want to say PCA had a silhouette score of about 0.4 and t-SNE had a silhouette score of about 0.6. And so the clusters that it was able to identify were much better than PCA. And so a silhouette score, goodness of clustering, there's a couple other methods you can use, but those are the two most popular ones that you can use to actually more quantitatively compare them.